Hello NEET aspirants, welcome back to BioBoost. Today we will go through 10 important MCQs from molecular biology. Stay tuned till the end of detailed explanation that can help you score higher. First, question number 1. In split genes, the coding sequence are called option number A, exons, option number B, introns, option number C, cistrons, and option number D, operons. The right answer will be exons. Now the explanation. Look at this picture. In DNA, we see that DNA is composed of exons, and this is which I marked in red. Marks in red. It is exon part, which is expressive part. Expressive. And in between the exons, there are present the introns, which is inactive part or which does not carry any protein coding sequences. So, split genes have both coding and non-coding sequences. Coding sequences or expressive sequences is called exons, and non-coding sequences or inactive sequences are called introns. And after the splicing, here these coding sequences get attached, and here these three coding sequences get attached. Various types of splicing can be happen, like this is exons are skipping the introns, and then ultimately from this type of expressive DNA, mutually exclusive exons during splicing can be happened. Mutually the exons can be spliced, and this type of DNA can be formed. Alternative splicings can happen. So the correct answer is the coding regions are known as exons. So they stay in the mature mRNA, means messenger RNA, and after transcription the exons produce the proteins. Introns, the non-coding regions, are spliced out during RNA processing. Okay. Now the next question is, it is said that in question number 2, RNA polymerase Okwa 1953, who discovered it, has polypeptide chains. So the question asked that how many polypeptide chain from the RNA polymerase holoenzyme. The options are given as A 1, B 2, C 4, and D 5. The correct answer is option number C 4. It is the correct answer. Now the explanation part. Here we are seeing the holoenzyme image of RNA polymerase. Here we can clearly see that here present two alpha protein chain or polypeptide chain, one beta and one beta prime chain, and one omega chain. So two alpha chains means one beta chain, one beta prime, and one omega chain. So total number of 4 polypeptide chains are present in RNA polymerase holoenzyme. In case of prokaryotes, in case of prokaryotes, RNA polymerase has 4 core subunits, that is alpha, beta, beta prime, and omega. Together they make up the enzyme responsible for transcription. Okay, so from DNA, when the after transcription, when the mRNA produced, this enzyme help in this total process. Okay, now our next question. Next question, in question number 3, it is given that central dogma of modern genetics is option number A, RNA to DNA to protein. Option number B is DNA to RNA to protein. Option number C is DNA to RNA to protein, and option number D is RNA to DNA to RNA to protein. So here the correct sequence of central dogma in modern genetics is DNA to RNA to protein. Now the explanation part. Here, look at this picture. It is the double helical structure of DNA molecule, and after replication it produces another strand of DNA, so it becomes a double helical structure, and after transcription it produced mRNA or messenger RNA, and after translation it produced protein. So the sequence is DNA to RNA. After transcription RNA is produced, and after translation RNA produce the protein. From RNA, during translation, the various amino acids get assembled and produce the protein molecule. So the correct sequence is DNA to RNA to protein. Okay, this describes how genetic information flows from DNA to RNA by transcription, and then from RNA to protein via translation. Now our next question. Our next question says that, question number 4, maximum formation of RNA occurs in. Maximum formation of RNA occurs in option A, cytoplasm, option B, nucleoplasm, option C, nucleolus, and option D, ribosome. And the correct answer is option C, nucleolus. Option C, nucleolus. Now the explanation part. Here, look at this picture. This is a picture of nucleus, and this is the part of nucleolus. Here we can clearly see that around the nucleus there are present many 40S or 60S ribosomes. As the ribosomes assembled here, and nucleolus function to produce the assemble line of ribosomes, and also the nucleolus is also primarily made up of ribosomes and rRNAs. So ribosomal RNA synthesis mostly happen in the nucleolus. Nucleolus is the prime site for rRNA synthesis, means ribosomal RNA synthesis. And also here we can see presence of multiple ribosomal subunits around the nucleolus, and it functions to assemble the ribosomes. Hence it is where the most rRNA is made, or rRNA is made. So the correct answer is nucleolus. Okay, now we are going to our next question. Question number 5. Question number 5, who synthesized the first artificial generated? Who synthesized the first artificial, this is only gene, artificial gene. Okay, so the options are given as A, Morgan, B, Nirenberg, C, Khurana, and D, Mendel. And the correct answer is option C, Khurana, or Hargobind Khurana. Now its explanation part. Hargobind Khurana first made the first synthetic gene in 1970, first synthetic or artificial gene. He produced the first synthetic gene in laboratory in 1970, and it is a breakthrough, big breakthrough in biotechnology. And for this he also awarded with Nobel Prize. He was also awarded with Nobel Prize. Now our next question is question number 6. Our next question is question number 6. Question number 6 says that products of one gene requires to activate another gene. Products of one gene requires to activate another gene is called, option number A, catalase, option number B, coenzymes, option number C, episomes, and option number D, transcription factors. Option number D, transcription factors. The right answer will be transcription factors, option number D. Now its explanation part. Here in this image we can clearly see that transcription factors, these proteins, binds to promoter region. Transcription factors, this protein binds to promoter region and helps RNA polymerase to initiate transcription. So this transcription factors is one factor, that is a protein which generates from a DNA sequence or a gene and helps to produce initiation to the other gene. Okay, now the next question is question number 7. It is said that in a DNA molecule cytosine is 18% and then the percentage of adenine will be option A 32%, option B 64%, option C 36%, and option D 18%. Then what will be the correct answer? The correct answer is option number A, 32% is the correct answer. Now there is two type of bases present in DNA molecule, that is purine and pyrimidine. Purine and pyrimidine. In purine base there is adenine and guanine. Adenine is represented as capital A and guanine is represented as capital G. And in pyrimidine base there is thymine, which is represented as capital T, and cytosine, which is represent as capital C. So in DNA molecule, in its double helical structure, in one strand of DNA and in another strand of DNA, there always pairing happens between the purine and pyrimidine base. So when A always pairs with T, adenine always pairs with thymine by 2 hydrogen bonds, and guanine always pairs with cytosine with triple hydrogen bond. So in question it is given that cytosine is 18%. So when cytosine is 18% means guanine also present in 18%, because they must be equal in number or equal in percentage. So here 36% already gone. So remaining percentage was 100 minus 36, which is 64%. So A plus T collectively made this 64%. So individually A percentage will be 64 by 2 percentage equal to 32%. So A is the correct answer, 32%. Okay, now we are going to the next question. Question number 8. Question number 8, it is says that which one is DNA nitrogen base with single ring structure. Options are given as option A uracil, option B thymine, option C adenine, and option D guanine. And the correct answer will be option number B, which is thymine. As we know that uracil present only in RNA, so it is not relevant for DNA nitrogen bases. And we also know that thymine, adenine and guanine are the purine bases, and thymine only here is the pyrimidine base. Now its explanation part. In this picture we clearly see that here purine bases, it is adenine and guanine. Here it is said that a heterocyclic aromatic compound, as heterocyclic because it is formed of nitrogen and carbon molecule, and it is the purine bases are formed with one pyrimidine ring fused with one imidazole ring. It is a pyrimidine ring and one imidazole ring. Here also in case of guanine, it is one pyrimidine ring and one imidazole ring. But in question it was asked that which nitrogen base has a single ring structure. In case of pyrimidine base, here we can see that in examples of pyrimidine base there are present cytosine, thymine and uracil. And in every base, in each of the base, there are present only one single ring, which is pyrimidine ring and pyrimidine ring. But in case of uracil, it is not relevant for DNA, so it is already excluded out. And in between the cytosine and thymine, in option only the thymine is given, so thymine will be the correct answer. Okay, now in next question, in question number 9, it says that operator gene of lac operon is turned on when lactose molecule binds to option A promoter site, option B mRNA, option C operator gene, and option D regulator gene. And the correct answer is option C, which is operator gene. Now we are going in the explanation part. Here we clearly see that this is RNA polymerase, and the first picture is in absence of lactose. In absence of lactose molecule, the RNA polymerase, in presence of RNA polymerase, the transcription is not happening because the repressor molecule is get attached with the operator, and RNA polymerase could not proceed reaction. But when the second picture is for the in presence of lactose. When lactose is present, RNA polymerase can proceed its action because repressor molecule get attached with the lactose molecule, and it cannot get attached with the operator. So RNA polymerase can easily proceed its action and can go for the transcription. in this way the lactose operate the lac operant site. Okay, now the next question is, which is question number 10. Question number 10, it is says that RNA code for DNA codon ATG will be option A ATG, option B AUG, option C USC, and option D TSC. So it is asked that which is the RNA code for a DNA codon. If this DNA codon present in the RNA, which will be its code? So here option B is the correct answer, which is AUG. As for A in RNA, it will also remain as A. As for A in RNA, it is also remain as A. But as for T, as no thymine base present in the RNA, it will represent it as U, uracil, and for G, it is represented as G also, because guanine also present in RNA. So AUG will be the correct codon for RNA, for DNA codon ATG. Now its explanation, we are going for its explanation. Now see that these two are the DNA strand, and here the one strand DNA sequence, and it is the other strand DNA sequence, and this is transcription happening and producing mRNA. In mRNA, T is represented as UA. So A present in mRNA, but in case of A, it is represented as U. So there we can see no presence of T in the mRNA sequence, no presence of thymine, but it is replaced by U or uracil. So AUG will be the correct answer for this question. Thank you. Hope this session is good for you, and if you like this video, share with your study buddies, and stay tuned and stay with us for more attractive video in future. Okay, thank you.